Hello, everybody, and welcome to today's lecture. So this is on sensory systems. It's an introduction to sensory systems. Note that initially we were going to do sensory systems and vision and bioluminescence. All of that is too much for one day, I think. So what I'm doing is giving you the just the sensory systems video background on sensory systems. So you'll watch the video on that, we'll discuss it, and then I'll also present some of the vision material in class when we meet this week. So just realize this is not sensory systems and vision and bioluminescence, it's just the background on sensory systems. So please prepare your questions based on this, and then we'll take those plus some new material on vision when we meet in class. So here we go. Sensory systems. I like to start by introducing sort of a German idea. Germans are very useful for thinking about things in grand fashion. And so there's this idea of Umwelt. And so when you think about an Umwelt of an organism, you've got, the organism has sensory modalities, right? It's got mechanisms and pathways whereby it detects the environment around it and has some response to it. And you can think about this in a lot of different ways, but if you sort of take all those sensory modalities, that sensory information input, and think about what it is to the organism, that is its Umwelt. So it's sensory perception of its world, right? So you can think about this at any scale of any organism you want to. There's some ability to perceive the environment and respond to it, whether that's through movement, whether it's through biochemistry, changes in biochemistry, whatever it is, so whatever's possible for the organism in question. There's some detection of environment and then response to it. And we think about the sensory modalities that contribute to that. It creates this Umwelt. So the sensory perception of the environment. So I say here that any given sense may be important to an organism, so whether it's vision to a Hyperion amphipod, which is down here with these massive eyes, or not another, this ctenophore doesn't have any vision. You can scale this down to anything you want to talk about, microbes, fungi, whatever it is. There's some perceptive piece, and there could be a biochemical response, it could be a movement response, it could be some other physiological response. But there's still some perceptive sphere around the organism. And it may be that one sensory modality is involved, or maybe not. It just depends on the organism. Not everything is the same. And so I think that's an interesting concept in terms of sensory systems that reminds us that just because we're humans with a pretty good visual system, that's not the way other organisms experience the world. And so when you think about an organism and you're trying to understand how it senses the environment, then you have to think about what its capacities are, not just bring your own sensory bias into the equation. So with sensory systems too, sensory signals are processed in what are termed parallel lines. So you've got parallel organization of sensory information. And so think about it this way. If you have a peripheral nervous system, PNS, and a central nervous system, or CNS, where you've got information being processed, you've got that information's coming in through various mechanisms. It could be neurons that are bringing in light information. It could be neurons that are bringing in sound information, neurons bringing in touch information, neurons bringing in odor or smell information, taste as well, which is variant on smell. And so all of that information is coming in to the central nervous system along parallel streams. And the information that you get in one stream could be neutral, antagonistic, additive, or synergistic to these other streams of information. So for example, you bring in some bit of light information, that could be influenced in a positive or a negative way by the smell environment. So lots of ways these things can be placed together, neutrally, negatively, positively, or more than positively, right? So when you get two, so that's synergistic. When you get the addition of 1 plus 1 is more than 2. It's actually 3, 4, 5, 6, whatever it is. Yeah, Mom.
When you say like in parallel, you mean at the same time?
Yeah, so they're separate neurons, separate conveyance mechanisms. They can be at the same time or in sequence, but yeah, parallel in time and space. So when we think about again these modalities of the sensory system, an important thing to realize is that sensory cells are there to transduce a stimulus energy into something that the nervous system can deal with, which is an electrical signal. And so think about it. You've got a physical modality, whether it's touch, whether it's light, whether it's chemical. There's some physical or chemical feature in the world, and that has to get moved into an electrical signal, right? And so that's what sensory cells do, is they transduce a physical or chemical stimulus into an electrical signal. And there are receptors that are present for various sensory modalities, and this table just kind of goes through that. And up here, you're seeing two types of transduction mechanisms that you see on different sensory cells. So you see ionotropic transduction and metabotropic transduction. So with ionotropic transduction, you've got the binding of a compound opening directly ion channels, then ions flow through those channels, as opposed to the binding of some sort of molecule to a G protein coupled receptor, which then sends out a second messenger, which is happening here. And then so you've got a G protein moving and activating this production of cyclic AMP, which then opens up an ion channel. So this is an indirect, it's a cascading effect with a metabotropic receptor. I'll talk a little bit more about those receptors as we go. But if you look down here at this table, these are various receptor types, the stimulus that's perceived, and then the mechanism of transduction. And so if you look just at the top, mechanoreceptors are involved in touch, pressure, and proprioception. These are ionotropic. Right? If you look down to what some metabotropic, so taste and olfactory receptors, various tastes, smells, gases, chemicals from a distance, all use metabotropic signal transduction. Photoreceptors, light is also metabotropic. And so these mechanisms of transduction tend to be tied to the types of stimulus modality that you are interacting with. An interesting way to think about this, though, is that these any of these signals can be transduced, sometimes independent of what they're supposed to transduce. So, for example, if Cassandra punches Amanda in the eye,
No, sorry.
what's going to happen? What do you think is going to happen to Amanda's eye? Right. So there's light that's coming in that's generating a visual response. But if you've ever bonked yourself in the head or got hit in the face, you're seeing stars, you're seeing patterns. There's a mechanical disturbance that hits your retina when the lens gets pushed to the back of the eye, and it physically moves ion channels open. So you can actually open ion channels the way they're supposed to be opened through this chemically mediated metabotropic transduction, or you can physically pull the ion channels open accidentally by pushing in on them, right? So that's what's happening when you're seeing stars because of an impact. So you're actually getting a mechanical stimulus that's activating photoreceptors. So there are ways to kind of cross these lines, but this is the way they're supposed to function. But it just reinforces that there's a physical stimulus that's being transduced, and you can actually bypass that the way they're supposed to work by situations like that.
Well, I have a question about some taste receptors.
Yep.
Well, how come the different flavors are on different mechanisms?
So how come?
Salty and sour, and then sweet and bitter are different, but their taste receptors shouldn't is there really
Yeah,
that big
I mean
difference?
it's the same. You could ask the same question about color and vision, right? Why do we see color, right? Why do we taste gradients of flavors, right? So there are evolutionary explanations, whether they're correct or not. There are evolutionary explanations about why have different tastes, because you see they're not present in the same ratios or at all in all animals even. There's a lot of many organisms that smell and taste, and whether you smell or taste or do both. So there isn't sort of a good explanation. I mean, I think there are plenty of explanations about why you would have variation in those taste receptors, but I think it would be the same explanations as to why you would see color.
Oh.
It gives a more refined way to break up that sense and help you
piece together information about that sense. So if you think about sort of a sequence of events that happens with sensory systems, there's an input here, which is your total energy stimulus. So this is what's going to happen. We call this whole thing a reflex arc, what I'm going to draw, or what's going to pop up. We call it a reflex arc. So you bring in sensory input. There's some peripheral filtering. If it's enough of a stimulus after it gets through the filtering, then you start to transduce that signal, which has absorption, transduction, amplification, integration, and spreading of a signal. You then, if it's adequate enough after spreading, you have a receptor potential that gets generated. And then you encode that receptor potential or changes in it with spikes. So these are things like action potentials, which we'll talk about. So you generate afferent action potentials, which is on the bottom here. And then you have some output to the central nervous system. So you've got input to the receptor cells, output to the central nervous system where the information is processed. And then the central nervous system can send a signal out, which is efferent control, back to here or here. So you're signaling back onto the peripheral filtering or the transduction, which can affect how those mechanisms work. And I'll give you some examples of this. But that's a reflex arc. So it's afferent sensory input and efferent motor control output. We talked about afferent and efferent with circulatory systems too, where we talked about gills. So, and when I was talking about an adequate stimulus, so you're bringing in the sensory information, I talked about an adequate stimulus. Think about it this way. If you've got receptor activation on the y-axis for these two different functions, and you've got wavelength here, so this is how light works as a signal, and these are how chemicals work as a signal. Light is really nice because you've got these very ordered receptors that detect different wavelengths of light. Light itself is electromagnetic radiation that's organized by wavelength. Maybe that's why I like it so much. It's very ordered and tidy, right? But then you get into something like chemicals, which can differ in terms of molecular weight, electronegativity, the number of carbons, the length of the chain, which is tied into the number of carbons. So when you think about all the different ways that you could organize chemicals and the perception of those chemicals and what the sensory receptor could actually be sensitive to, it gets really complex. And so you can have a very neat and tidy system like light or a very disordered or complex system like chemicals. But in either case, you're going to need to have the adequate stimulus to detect it. So if you have a short wavelength or a UV sensitive visual pigment, it's not going to be able to detect light that's far red light. Or if you're sensitive to very small molecular weight compounds, and you've got in front of you a very large molecular weight compound, then that is not an adequate stimulus. And so the stimulus has to be appropriate to make it to the next step, right? So now, in terms of sensory transduction, I'm just putting up one way that light is perceived. And so this is just an example of a photoreceptor. And so you've got all of these molecules that are involved in that G protein cascade, basically. So you've got the light coming in right here. And there's a whole series of molecules that are involved in actually transducing that light signal. It ultimately makes it so that there's a physical movement of the membrane. And then that's where the ion channels are, and that's what's actually opening this thing up. And then there's a whole series of enzymes and other cellular signaling that's involved in actually shutting down that signal as well. I'm not concerned that you know all of these steps. I'm just concerned to make the point here that this process of absorbing the signal, bringing it in, transducing it, amplifying it, integrating it, and spreading it can be fairly complex. And it involves molecular machinery that's often, so in the case of photoreceptors, it's all sort of in the membrane of the photoreceptor. Ultimately, you're generating a receptor potential. And what should happen there is that you'll ultimately will make a spike encoding mechanism from that receptor potential. And so if we look up here, this is the excitation of a stretch receptor in a muscle. So when you stretch a muscle, the way that so think about it this way. If you stretch your arm out, and you're stretching your arm out, you can feel that your arm is stretching. Give it a try. Stretch your arm out. Now lift it up and up and up and up. You can feel that your muscle is stretching, right? You can feel that it's stretching. Then bring it down. It doesn't feel that it's stretching anymore, right? So what's happening as that muscle in your arm stretches, action potentials are being fired that gives you a sense of the magnitude of that stretch. So the actual height of the action potentials is always the same in terms of membrane potential in millivolts, but it's the frequency of them that's changed. So this is called a frequency encoding mechanism. And so if you stretch at increasing amounts of intensity, you're seeing more frequent action potentials. And so what's happening as you stretch that arm more and more and more, there are higher frequency action potentials that are getting into your central nervous system and telling your brain that you're stretching your muscle. Or what you can do is have a depolarizing signal. And so in this case, this is showing you a light flash, and this is membrane potential as a function of time from low to high intensity light flashes. So what you're seeing is these drops in membrane potential are getting more and more magnified when it's a bigger and bigger light flash. And so it could be the magnitude of the depolarization that gives you a signal, and then you end up with an action potential. So there are ways to have spike encoding and then ways to have just depolarization signals that ultimately will get brought into an action potential. But still, it's conveying information. It's conveying magnitude of the response, magnitude of the stimulus. And so what you've got here is just an example of efferent control and filtering. And so we'll just these so it's a little clearer. And so what you've got in this situation is an example we did a few years ago where you take a little squid and you immobilize it in a piece of tubing and you let it swim and do its squid things, and then you give flashes of light of different light wavelengths. So starting in the ultraviolet, violet, blue to green, yellow, and red. And what you're seeing in this little section here, this right here, and here, here, those are the pupils. So this is the pupillary response in a squid. And so if you look at what's happening, you can see, so here's the little pupil right here, that little lid that comes down. You're constraining the pupil aperture by a lid that's going to drop down in front. And so as the wavelength changes from sort of this violet, not much of a response, to blue and really nice response here, where this is a little slit now. And then you see back here into the red, there's not much of a pupil response. And so what a pupil response is, in a squid or in you, is an efferent motor response that's going to knock down, peripherally filter the light coming into the eye, right? So it's a way to control the amount of the signal getting in so that you can perceive it better. Yeah, Amanda?
Is the pupil the black part?
The pupil is the black,
Okay.
and then the little lid coming down is the silver part.
Okay.
So it turns into sort of a U shape when the lid comes down, in this case. Yep, Nick?
You might have just been saying this, but how? So, like the lid doesn't block off. It would just lower the amount of light, but you still see the same amount. Like, why would you do this instead of like? I mean, I guess it's not an option, but like instead of like dilating pupils, why would you have something cover half of your pupil?
Yeah. So, well, dilating is going to be to open it up. So it's the opposite of this phenomenon, right? But
yeah.
But I think your question is, yeah, why would you knock down the stimulus coming in?
Yeah,
so
to
the reason for that would be that the way that those sensory receptor cells work, no matter what the modality is, they can be they can be flooded with signal. So they work over some range of signal intensities, and when you go too low, you're beyond the limit of detection, and when you go too high, you're beyond the limit of detection. So you can't detect it. There's sort of an upper and a lower limit, I guess I should say, where you're not going to be able to transduce the signal. And so what this does is it knocks down the incoming signal so that you can then effectively measure it.
Okay.
Okay, so putting that to like another example, just to make sure I understand what you're saying. It's like when the sun's like shining super bright and we kind of like squint our eyes so we can see better. Is
Yep,
that
you're
like
doing-
that
yeah.
same thing? So you're like adjusting.
Yeah, you're doing, and without you knowing it, your pupils are already doing the same thing. So there is, your central nervous system has efferent control over some things like sort of dialing down the pupil so that you're limiting light coming into your eye, but that doesn't necessarily give you enough control, right? So you're sort of squint a little bit as well, or put on sunglasses.
Okay.
So then you're overriding, you're doing above and beyond the peripheral filtering that your body can do.
Okay.
So that's pretty common with sensory systems. So I've talked about neurons a little bit already. Neurons are, for animals at least, sort of the mechanism that this information is conveyed from place to place in the nervous system. So those would be neurons. These are just 3 examples: a retinal bipolar cell, a retinal ganglion cell, and a retinal amacrine cell. So you've got different types of structures, look like little trees in some ways. You've got dendrites at the top, you've got a cell body, and you've got the axon. And so sort of no matter where you're looking at whatever type of cell you're looking at in terms of a neuron, you're going to see these elements, but they may be more elaborate in some cells or less recognizable in others. So for example, in the case of a retinal amacrine cell, a really sort of elaborate arborized group of dendrites up here with a little axon and a cell body, and the axon's kind of like going backwards on itself. It's not this sort of classic dendrite to cell body to axon arrangement. So there are lots of ways to organize that. This. Or I can't because it's stuck. So these are neurons. But the other thing that you have, which are incredibly important for how neurons function, are called glia, or glial cells. And so glia are oligodendrocytes, which is what's missing here. Oligodendrocytes. You see oligodendrocytes in the central nervous system, Schwann cells, which are in the peripheral nervous system, and astrocytes are around capillaries. And so what these are support systems. So astrocytes, oligodendrocytes, and then these are microglia, which worry about. Schwann cells you won't see here. They're in the peripheral nervous system, but they're like oligodendrocytes. And so what these are basically ways to, they're metabolic support for neurons, because neurons are pretty busy doing their conveyance mechanisms, and so the cell bodies are often reduced and don't necessarily have the metabolic machinery to power the cells. And so it's common to have glial cells that can provide that metabolic support, as well as provide insulation. So if you look at Schwann cells and oligodendrocytes, you're seeing this wrapping. See, there's sort of wrapping around the cells. So they're insulating it like you would insulate a wire. Because these things are ultimately, the neurons are ultimately conveying electrical signals. And so there's going to be loss of ions, and we'll talk more about that in a little bit. But these cells, the glial cells, are providing that insulation. Bless you.
Thank you.
So let's talk a little bit about resting membrane potential. So we want to talk now about how those signals are being conveyed, right? So in order to think about how the signals are being conveyed through a nervous system, we need to first talk about what's happening to a cell in terms of its membrane potential. And so in order to do that, we have to define a couple of things. We have to define current, which is the net movement of electrical charge, as compared to voltage, which is a separation of electrical charges. And so if you have a voltage, you have a separation of electrical charges that can do work when it's allowed to flow as a current, right? So once voltage is allowed to flow as current, you can do work with that. And so it turns out that squid have been pretty important for how we understand nerves to function. They've been a model, and the reason for them being a model is they have giant axons. And we'll talk about why they have giant axons when we talk a little bit more about glial cells in terms of myelin. But they've got big axons, so you can take out a little chunk of it and you can stick it in a bath and you can squeeze the ends so that the juicy insides, the ICF, doesn't come spilling out. And what this is showing you is a voltmeter, like you would have in a house. You can do that too. You can also just have two little electrodes. So there's a reference microelectrode here on the right, this little tube, and then on the left, a recording microelectrode. And this is just showing you that if you take a voltmeter and you have the recording and the reference electrode in the bath outside of the neuron, what's the reading going to be on the voltmeter? It's going to be 0 millivolts, because the electrodes are in the same place. If you then go over here to this side, and you were to drop the recording electrode inside the neuron and have a reference electrode outside, you would end up with about minus 65 millivolts on the voltmeter. The inside of the neuron is net negatively charged. The outside is net positively charged. And so what this is showing you is a resting membrane potential. And so resting membrane potential, the neuron is at rest. It's not doing anything. So what I want to spend a few slides then on is explain why when you take an electrode and you move it into the neurons, you get a resting potential of negative 65 millivolts. Like what's happening at the cell that's actually doing that. So this came up a few meetings ago. I think it was Hannah who mentioned it. This idea of sort of ions moving across membranes, and is it down a concentration gradient or what's happening? So we talked about this when we talked about just what happens, like why does chloride leak out of the cell, I think might have been the context for it. But that's going to come back into play here. So I want you to remember that if you have a concentration gradient, you're going to move from high concentration to low concentration. If you have an electrical gradient, opposite charges are going to attract, right? And so those two things are the physical reality of the situation. If we then set up a little thought experiment where you've got a box and you've got your voltmeter again with an electrode in either side, and you've got 1 millimolar of potassium chloride on either side of a membrane that's permeable to potassium, what's going to happen? Well, there's no net flux of potassium because it's the same concentration on both sides, and potassium has equal concentration on both sides. So there's no concentration gradient and no electrical gradient. And so everything's the same. But now if we set up a situation where we had 10 millimolar potassium chloride on the left, number 1, and 1 millimolar potassium chloride on the right, in box 2, initially what's going to happen is there's going to be a net flux of potassium from box 1 on the left to box 2 on the right. Why is that?
The concentration gradient?
Exactly. It's just a concentration gradient. And the membrane is selectively permeable to potassium. Right? So now, once you're at equilibrium, what's going to happen is you have, in this case, a flux of potassium from the box 1 to box 2, but it's not going to all move out to equilibrium, because as those potassiums move from 1 to 2, you're going to develop a positive charge on the box 2 side of the membrane when those ions flow through. And there's going to be a point when that positive charge, which sets up an electrochemical gradient, balances the concentration gradient. Right? Yeah.
And that's because it's selectively permeable, right?
It's because it's
the scale
selectively
is staying on
permeable.
the
Exactly. It's because it's selectively permeable. Yeah. And so you end up with a situation where it's actually not going to be like this case of having the same concentration on both sides. You're going to have a higher concentration on one side than the other, and a membrane potential that develops. So let's go back now to our cells. So we said here's our basic cell. We've got high potassium, we've got low sodium and low chloride on the inside of the cell. On the outside of the cell, we've got low potassium, high sodium, and high chloride. If we just sort of transition this sort of generic cell to a nerve cell when it's at rest, which is shown here in the upper right, you've got a sodium potassium ATPase. right? You've got a sodium-potassium ATPase, which is pumping in 2 potassiums, kicking out 3 sodiums. We've talked about this ad nauseum. You have a potassium channel, which is an efflux channel, so you've got a slow leak of potassium outside the cell. Let me just cue you in on this. You have a semi-permeable membrane, which is semi-permeable to potassium, just like that last slide. And you've also got a slight influx of sodium through a sodium channel. So there's a slight leak here. There's also some negatively charged protein on the inside of the membrane, a little bit more on the inside than it is on the outside. So if that's your nerve cell at rest, anything else I want to say about that? Blah, 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 blah. So think about what's going to happen here. You're constantly bringing in sodium, you're constantly bringing in potassium, constantly pumping out sodium. That's setting up a situation where you're going to have a slight influx here of sodium and this efflux of potassium. So that, in a lot of ways, is a lot like this, right? There's a lot more potassium flow than there is sodium flow. And so you're basically having this be your neuron, where there's high potassium and there's going to be movement that way, right? And so what's going to happen is you're going to have potassium leak out of the cell until it reaches electrochemical equilibrium, until the concentration gradient balances the electrical gradient, just like on that last box. And so what we call that is the equilibrium potential for K+. So the equilibrium potential. So the equilibrium potential for any ion is, you can just sort of define it as the membrane potential that develops when the concentration gradient is balanced by the electrochemical gradient. So in this case, there's going to be an equilibrium potential for K+, and there's going to be an equilibrium potential for Na+. And together, they're going to give you the resting potential. So again, the equilibrium potential is not the membrane potential, it's a portion of it, attributable to a single ion. So I'll just show you some examples of that. So the equilibrium potential for potassium and the equilibrium potential for sodium are shown here. This is called a Nernst equation. And so, has anyone seen the Nernst equation before? 1, 2, yep, somewhere. Yeah, there's another one called the Goldman equation, which is basically multiple Nernst equations stuck together. But the Nernst equation is showing you a squid. So this is showing you the Nernst equation for a squid neuron at room temperature and pressure. And so EK plus is 25 times the natural log of the concentration of potassium on the outside divided by the concentration of potassium on the inside. And that's going to equal 25 times the natural log. For a typical squid neuron, 20 millimolars on the outside and 400 millimolar on the inside of the neuron. Remember, the inside is a higher concentration of potassium than is the outside. And you end up with about negative 75 millivolts as the equilibrium potential for potassium. So that's the membrane potential that develops so that this concentration gradient matches the electrical gradient for these conditions. If you do the same thing for sodium, 25 times the natural log of sodium on the out over sodium on the in, 25 times the natural log of 440 millimolar over 50 millimolar, right? So what you've noticed about the potassium and the sodium is that the numerator and the denominator are flipped. And so now this gives you a positive 54 millivolt membrane potential, right? So equilibrium potential for potassium and equilibrium potential for sodium.
Where's the 25 coming from, or is that just part of the equation?
It's if you reduce down. So I've taken the room temperature and pressure parts to make it simpler. It comes from basically this is a temperature-mediated phenomenon and pressure-mediated, and so I've reduced all of the room temperature and pressure bits down to a value of 25.
Okay.
So this is variable depending on these values.
Yep.
Yep.
So do we not too much care for chloride?
It's a good question. It turns out that you don't have to care too much about chloride. There's a slight efflux of chloride, but it turns out you don't have to care about chloride all that much to generate the resting membrane potential. So chloride matters, but in terms of setting the resting membrane potential, it's primarily sodium and potassium, because it's those ATPases that are really setting up the major ionic gradients. Chloride is there, but it doesn't do too much. The Goldman equation, which I mentioned, you can include chloride. So if you did sodium, potassium, and chloride together using the Goldman equation, you could get a resting membrane potential, but it doesn't give you all that much more information. You can get pretty close to just paying attention to sodium and potassium. Good question. Anything else I want to say about that? So if you think about a squid membrane potential, so Vm is about negative 65 millivolts. What does that tell you about the relative role of potassium and sodium in setting the membrane potential, Vm? Which is more important?
Potassium.
Potassium, yeah. Because it's pretty much giving you the membrane potential pretty close, right? It's pretty much, the membrane potential is pretty much set at the equilibrium potential for potassium, but not quite. It's slightly less negative, which means there's a role for sodium as well in making it slightly less negative than it would be otherwise. But it's primarily the permeability of the membrane to potassium that sets the resting membrane potential. Right? You can also sort of show that experimentally. You can knock out those potassium channels, those potassium efflux channels, with a couple different ways. There's a couple things you can do experimentally. You can knock out the sodium-potassium ATPases with something called wabane, which just poisons all sodium-potassium ATPases. Not a really great thing to work with because you have a lot of sodium-potassium ATPases, and so it's very dangerous to work with. But you can just stop all of that activity. And then if you're not pumping the sodium and potassium anymore, you basically have a membrane potential that just goes flat. So it shows you that the sodium-potassium pump is really important for that. But you can also take potassium acetate and just put it in the medium and increase the extracellular concentration of potassium. And then you can, so that's this one up here. I was pointing to the wrong one. So if you increase this concentration of K plus on the outside just by putting potassium acetate around the cell, that could be increased. And then you can make that membrane potential go to zero, basically, if you make that 2400 instead, right? Or a higher value. Yeah.
So are the changes in like membrane potentials perceived to us as like stimulus?
Yeah, so the changes in membrane potential are perceived to us as action potentials or depolarizations of the membrane, which, depending on what you're talking about, if it's like a retina, hyperpolarization or depolarization from, and I'll talk about what those terms mean, from the membrane potential are perceived as signals in the nervous system. Yep. So this is if you're going to spend money, you have to have money in your wallet, right, to spend. And what's happening is, or credit to spend. But in this case, the VM of negative 65 millivolts is basically the starting point. That's how you start spending information in your nervous system, right? So this is what you start from. And then if you deviate from this, becoming more negative or less negative, then that's a change that can be perceived in the nervous system, right? You're spending that membrane potential. So it's deviations from that we're going to be concerned about. So really to that point, hyperpolarization, your membrane potential becomes more negative. Depolarization, your membrane potential, your resting membrane potential becomes less negative. So I was talking about these just a second ago. And if we think about a typical nerve cell at rest, the outside of the cell here and the inside of the cell, we've got a high concentration of sodium on the outside, a low concentration on the inside. Here I'm saying VM at around negative 70, but it could be negative 65, negative 70, whatever it is. What that's going to mean in the case of sodium is that there's a concentration gradient that would favor sodium movement into the cell, and there's an electrical gradient that would favor sodium movement into the cell. And so all that would need to happen is to open up a floodgate of sodium channels, and then you would move sodium down its concentration gradient and its electrical gradient. And so that's basically what happens. And I'll show this to you, but this value of Vm is going to tend to go to the equilibrium potential for sodium at that point. Because now, as Leandra mentioned, it was the equilibrium potential for potassium was what set the Vm to begin with. If you then make it so that the equilibrium potential of sodium sets where you are in terms of membrane potential, it's because the permeability was drastically increased for sodium to the point where permeability to potassium doesn't matter, right? And so let's think about how that happens. So that's an action potential, basically, is what we just described. So you've got membrane potential here, starting, let's say, at negative 65 millivolts. And what we're going to describe is this sort of functional form with a change in membrane potential over a change in time, where we have a temporary depolarization and recovery of the axon membrane. The height of this thing, you'll notice, this is showing it as around 40. It depends on the concentrations of sodium, but this is basically stopping at the equilibrium potential for sodium, whatever that is. I said 58 for a squid neuron. This is showing it a little bit lower, but that's where that maximum value comes from. It comes from the equilibrium potential for sodium. And so what's going to happen is you have a rising phase, an overshoot, a falling phase, and then an undershoot, and then it's going to come back into where it started. And what we can do is we can talk about what's happening in these different phases. Notice there's a voltage threshold here and a resting potential line here. So we start out at a resting potential. We depolarize the cell to the point where it's enough to surpass a voltage threshold, and then an action potential is fired. And then there's a recovery that comes back into place to get you back to the Vm. So one thing that's useful about this, or useful to think about, is to know that these are all or none. So an action potential is all or none. You don't have half of an action potential. You exceed the voltage threshold and you generate the exact same action potential. So recall when we looked at those muscle stretch receptor little traces, and you had the same height of an action potential, just more frequent versions of them. That's because an action potential is an action potential. There's no bigger or little action potential. If you exceed the threshold voltage, you fire one of these things. And that's information. So this slide is just breaking out the resting membrane potential and how it's developed. And on the right, it's showing you the equilibrium potential for potassium, the equilibrium potential for sodium in this particular case, and where we are in the action potential. So that goes along with what's happening in this little diagram of the neuron. So a resting membrane potential, as I said, was basically set up by the potassium leak channels. So there's a potassium leak channel that's open. Potassium is moving through that leak channel according to its electrochemical gradient. There's a voltage-gated sodium channel, and there's a voltage-gated potassium channel. That's what's happening at rest. The permeability, the P of K, permeability of potassium, is greater than the permeability to sodium. See that? So we're all clear on what's happening at rest? Yeah? Newark, clear? More or less? Okay. The rising phase. The section here. So we've exceeded the voltage threshold. We're firing an action potential. What's happening in terms of this is the voltage-gated sodium channels are set to open when voltage exceeds that threshold. So when you exceed that threshold voltage, all of your voltage-gated sodium channels open up. And now all of a sudden, the floodgates to sodium have opened. The permeability to sodium is far greater than the permeability to potassium. And you start to depolarize the membrane towards the equilibrium potential for sodium.
Okay, I got a question.
Yeah.
Right now, okay. So when you say like action potential, like going back to the arm thing, so we're talking about what's going on in our neurons or whatever when we lift our arms?
That's sending a signal from the neuron in your arm
The signal.
to your brain that says, stop stretching my arm.
Okay, and then now we're talking about what these neurons are doing when they're at rest.
It's the mechanism. So what we're talking about is the mechanism that creates that signal that's then going to the brain or whatever the central nervous system is to say.
So these are the
That's-
mechanisms that are creating the signal.
Yep.
Now, why exactly is it just like potassium and sodium? Just like, is it just like those are like just what's in there, or just like?
So those are the major ions that are pumped back and forth across the cell, and that they set those ion gradients,
Okay.
the electrochemical gradients. They're setting membrane potential, and they're involved in the osmotic concentration as well. So
Okay.
they're used for volume regulation, which is what we were talking about the past few times. Those are the major inorganic ions.
Now, when you talk about, what did you say, mechanism potential? Is that, or
Membrane potential?
membrane potential, that's what I meant. I probably should have asked a couple slides ago.
That's fine.
But like potential, so going back to the arm, is that how high I can lift my arm?
No.
No.
It's the actual cell membrane. So imagine this. So if we go back to this situation, let's go to this one. So if this is the cell membrane,
Yes.
we just plucked a bit of the cell out, and
Yeah.
it's in a dish now.
Okay.
It's a long cell, right? This is all one cell. It's a long
Yeah.
cell. And so we just plucked a little section of it. And we take an electrode and we place it within that membrane.
Okay.
And imagine these are sealed off, so there's actually no flow in and out. Then the inside of the cell is negatively charged relative to the outside of the cell.
Okay. I get
That's what
that.
these little pluses and minuses are showing.
Now, potential for what?
So then that gets into the separation of charges. So it's a measure. So it's the VM, the voltage. It's a potential to start charges to flow as current. So the terminology in sort of the physiology literature
Yes.
is to call it a potential because it's a voltage, which is a separation of charges that can become current when you let them flow. So it's
Okay.
the membrane potential.
Okay, so it's
It's
potential
a voltage measurement.
to be a current.
Yep, it's potential to be a current.
Now, when it's a current, like, what's,
Then it flows and you have some value for
It just
the current itself.
and like, it's like this current, it's like, what's the cloud? What's the current? Do we need to like meet up later or something?
Yeah, we can talk more about it for sure. Don't get confused with the terminology, right?
Too late.
So the resting membrane potential
Okay.
is just the voltage, the separation of charges across that membrane.
Okay.
And so we it's all relative to a reference, right? So if this outside changes or some permeability changes, that's going to change the membrane potential, the separation of charges present at this membrane,
Okay.
right? And then what I went through after this was talking about how you actually quantify this.
Okay, yeah.
And the way you quantify it is based on what the concentrations of the ions are on both sides and how permeable the membrane is to the ions.
Okay. Can we talk like
Yeah,
after
we
class?
can go
Just
through
like
that.
for like a
Yeah.
hot second after class.
Sure. That's fine.
A little bit.
Yep. So where were we before?
Hannah has a question.
Oh yeah, Hannah, go for it.
Sorry, so what actually allows you to exceed the voltage threshold?
So there are ion channels all over, particularly in the dendrites, where if you open up those channels because, let's say, a neurotransmitter bound to them, that's going to open up sodium channels, which is going to depolarize the cell. So we had talked a little bit. There were, it was Matt's presentation during the slides, during the bibliography slides about GABA receptors. So that's another example of a channel where you've got a voltage. Well, in that case, it's a gamma aminobutyric acid channel. So there's you bind a neurotransmitter, GABA, and it opens up the channel and lets chloride ions flow, right? Really, it lets any negatively charged ion flow through it. And so if you think about neurotransmitters, those are acting, and I have some slides on this later too, talking about synapses at the dendrites, which are going to change the polarity. They're going to increase the permeability of that cell to sodium. So it's not just the voltage-gated channels that are doing that. This is how you generate an action potential. But if you take a step back and say, okay, what's your question? How do you actually get to the rising phase to begin with? How do you go over that threshold? The way you do it is by depolarizing the cell at other parts of the cell, and then sort of aggregating that depolarization at the cell body, which is where the decision to fire an action potential rests. So it's the aggregated depolarization from the action of neurotransmitters around the dendrites, really, that do that depolarization. And if that doesn't make sense to half of you, or 3/4 of you, or all of you, then it'll become a little clearer when we look at some of the later slides. Good question. So let's just take a step back and go back to the sodium channel. So during the rising phase, you'll notice now that the voltage-gated sodium channel was closed. Now it's open. And that's what's causing this tendency for the membrane to move towards the equilibrium potential for sodium. That's great. Your voltage-gated potassium channels are still closed. That eventually gets you to here, the overshoot. But then you're in the falling phase, where potassium permeability now is far greater than sodium permeability. So how does that happen? Well, you still have this efflux channel, which hasn't changed for potassium. But now, see this little ball that's popped into the little hole in the voltage-gated sodium channel? You've temporarily knocked that channel out. So it changes its conformation so that sodium can no longer flow through it. But the voltage has now opened up, this change in voltage going the other direction has now opened up potassium channels. So now you've all of a sudden massively increased potassium permeability, and this voltage is going to go down. Right?
It's going back to that,
It's going
the
back-
bottom, with
back
that, the
negative.
resting water?
Now because potassium permeability now is increased, and so now now the membrane potential is going to tend towards the equilibrium potential for potassium. Because this is the dominant ion that's moving now across that membrane. Right? So evolution can work on these steps, where you actually can have different timing of your what are called delayed rectifier potassium channels, which are what these guys are. So how quickly they work, the voltages that they work on, the time delay between these two, those are evolutionarily modifiable. And so for species or organisms and species that need to have neurons that function more quickly, these things will be modified to function more quickly. For ones that have very, very sensitive but very slow cell dynamics, these things can be more slow. So just an interesting thing to think about is that these aren't like a static fixed thing. There's some potential to
So
evolve.
just to make sure I understand. So sodium's flowing in, positively charged, and then once it gets to a certain point, it plugs up that
Yep.
transporter, and then potassium flows out
Yep.
because it's also positively charged.
Yeah, so it's potassium flows out because of the concentration gradient initially,
Okay.
and then it's going to set up, and then
But
also-
then backwards, does
Yeah.
that matter? So what happens at that point? Like, does it come back through? Or?
So you still have a net. So this is kind of showing you the charges are gone, but so there's a positive charge here. When this opens, this membrane potential is there's a higher concentration of. We ended last time talking about action potentials, which remember are these temporary depolarization and recovery of the axon membrane. You get this little waveform. We were then talking about what happens at these different steps in terms of the ion channels that are creating this this action potential. And so we said that initially, you've got these potassium leak channels, and they're just leaking potassium going down its electrochemical gradient. That's establishing the membrane potential that you're seeing with sort of a net negative on the inside, net positive on the outside, and a negative 65 roughly millivolt voltage. So that's fine. And then during the rising phase, when you depolarize, the sodium channels are opening. Question that came up was, how does that happen? We'll go into that more. It happens by basically the action of neurotransmitters elsewhere in the cell that are causing that membrane to change its polarity. So you you have an ion that binds. I don't want to point to this because it's a different sodium channel, but you have an ion that binds and it opens up sodium channels, right? You have a neurotransmitter that binds an ion channel and creates permeability to sodium, for example, calcium. That's going to depolarize the cell. So you've got this depolarization. If the depolarization is such that it is greater than the threshold for the action potential, you're going to fire an action potential. And then that action potential is going to move the membrane potential to the equilibrium potential for sodium. It's a lot of potentials, right? But voltage, you have an equilibrium potential for potassium, an equilibrium potential for sodium as your sort of end members. You're actually at somewhere sort of not in the middle, but skewed towards the EK plus, because that's where the resting potential is the VM, the membrane potential. Sodium channel's open, you depolarize the cell. The membrane potential moves past zero towards the equilibrium potential for sodium, because all that sodium is rushing into the cell. The sodium channel itself changes its conformation, which is represented by this little ball kind of swinging into the hole there. And so it basically makes it so that sodium can't pass. It's not recovered because you couldn't open it again. It's permanently, not permanently, it's immobile. It's not able to pass any sodiums through it for the moment. And it can't open up again. And at that point, voltage-gated potassium channels open up and then let the potassium, which is built up inside the cell, out. So then you're increasing the permeability to potassium, which then makes this, the membrane potential drop back towards the equilibrium potential for potassium. And again, on top of all of this, and not shown here, the sodium-potassium ATPases are just doing their thing. They're bringing in the potassiums and kicking out the sodiums. Right? So eventually, you've got all this potassium leaving the cell. You're going to have a little bit of what's called an undershoot. So you're more negative than the equilibrium potential for potassium. And then eventually, you'll get the equilibrium back, because these channels, the voltage-gated potassium channels, will close. Okay? So using the same kind of architecture, we've got these neurons, and the whole point is to send these action potentials down the neurons. And those are going to be the signals that are being sent from one place to another, right? And so, but the idea is that the membrane potential is going to move kind of as a wave along this change in membrane potential is going to move as a wave along the cell. And so one thing that you can think about is then, how does this happen? How does it move in a one-way direction? Because information generally, at least we used to think, moves in one way along neurons, from dendrites through the cell body and the axon, and then eventually to the next neuron. Turns out that that's not quite the case. There's examples of what's called a retrograde synaptic transfer, but that's way beyond what we need to worry about. But generally, information goes in one direction along a neuron. And the way that that works is you've got ions moving in here, and so you've got this sort of open depolarization area in the middle, right? Where ions are coming in. So you've got Na plus sort of making its way in. And what's going to happen is that Na plus, that depolarization, is going to move in both directions. But the upstream channels are inactivated, right? And so the sodium here, there's no, you can't change the membrane potential here due to the sodium. It's all being controlled by the potassium that's leaving. But the sodium can, when it's down here, can depolarize and start opening up these channels. And so because of this little temporary change in conformation of the sodium channels, you make it so that information can only move in one direction. Even though when you bring sodium in, it can actually diffuse in both ways. But you can't continue to depolarize on the upstream side. You can only depolarize on the downstream side because of that little inactivation. That make sense? So that's why information moves in one way in a nervous system along neurons. Let's see. Anything else I want to say about that? So this sort of terminology is called being refractory. So the neuron is refractory upstream of the action potential. And refractory is just sort of non-labile, right? So it's used in terms of sort of breaking down carbon or whatever it is you want to talk about being refractory. So this portion, the upstream portion of the neuron, because of this inactivation, it's refractory. And there's some refractory period before those little balls kind of pop out, and then the channel can be depolarized again. The voltage-gated sodium channel can be repolarized, re-depolarized. Make sense? Sort of. You're looking at me like you're crazy. So we've got action potentials. They're moving down neurons. You're losing some ions during this process because some of the sodiums are going upstream, some of the sodiums are going downstream. You're always moving ions in and out of axons. And so if you were to think about wiring in your house, there's insulation around that wiring, right? And so it's so you don't lose the current all along the way, right? That's why you've got insulation, so it can move down the wire and not jump off the wire. We want to talk about that, and we want to talk about it with respect to the speed of an action potential. And so I already mentioned that an action potential magnitude is kind of a fixed thing. It's similar among all animals. About 100 to 125 millivolts is the magnitude of an action potential. It's set by the equilibrium potential for sodium and potassium, right? And we said in the osmoregulation bit of the lectures that that particular aspect of ion concentration is set by what the ECF and the ICF can handle, particularly the ICF. So it makes sense then that if you're talking about the effects of that at the cells, then this is kind of a fixed thing. It's about 100 to 125 millivolts maximum, because you're talking about the equilibrium potential for potassium and the equilibrium potential for sodium. But what's very, very different among animals, at least, are conduction velocities. And so how fast the signal is being sent along a neuron. And there's various things that can affect that, with first and foremost the axon diameter. So the axon diameter. And there's two sort of relationships that we want to think about. V equals K. D is one of them, where this is velocity, and K is a constant, and D is the fiber diameter. Very annoying. And the other relationship is V equals K times the square root of D. Again, you've got K as a constant and D as the fiber diameter. And so this upper one is with myelin, and this lower one is without myelin. And I referred to myelin before. It's the glial wrapping around neurons. And so if you have a myelinated neuron, the velocity of an action potential, the speed at which an action potential is going to move down a neuron, is proportional to the fiber diameter. So the bigger the fiber diameter, the larger the velocity. So it's just a linear relationship with some constant. If you look and you don't have myelin, you've got V as being proportional to the square root of D. So you have to have disproportionately large fiber diameters to increase your velocity. You end up with something like a squid giant axon because they don't have myelin, so they have to get really, really big to conduct fast action potentials. I'll show you that in a graphic in a second. So axon diameter itself, so speed of the action potential varies with axon diameter for both myelinated and unmyelinated. The presence of myelin itself, glial wrapping around the neuron, is basically another aspect on top of that, and this is insulation. So myelin is insulation. You're insulating the neurons. So just by virtue of having it, it's going to go faster. The action potential is going to propagate faster. And then the third bit there is temperature. So temperature itself, you have faster speed of action potential propagation with higher temperatures. Q10s for this are about 2, like what we've talked about before. So you can sort of think about that. And if you think about what this means for sort of homeotherms versus other organisms, you end up with faster action potentials in homeotherms, so organisms that are maintaining a constant body temperature at an elevated rate with respect to the environment. So, what it allows you to do is actually have a smaller axon diameter and use the temperature effect to speed up your action potentials. So there are reasons, quote unquote, why this temperature effect is useful. So these things kind of play off each other. I'm putting them down here as like 1, 2, and 3, but they actually play off of each other.
Now with temperature, when you said with the axon diameter, could that be the same with the myelin, a higher temperature, and then, if there's no myelin? Or no?
So the relationship of axon diameter is affected by whether or not there's myelin, but a bigger diameter always gives you a faster velocity. So axon diameter itself matters.
Okay.
Myelin, with and without, matters. If you have myelin, it's faster. If you don't, it doesn't. The effect of temperature can apply with or without myelin, right? So myelin affects axon diameter, myelin affects the speed of the action potential propagation. So there's an effect of axon diameter, irrespective of myelin, doesn't matter. Whether you have myelin affects the relationship that velocity has with axon diameter, shown here. And temperature affects all of this. But I'm not sure that it affects myelinated versus non-myelinated axons differently. I don't know the differential Q10s. Yeah.
If there's an axon that typically has myelin, but like the myelin was like destroyed or damaged, can signals be lost?
Yeah. So muscular dystrophy. So the degradation of myelin is a medical condition, right? And so you can you lose muscle control basically. So you lose the signals can get lost. Yeah. So you're not able to send neuronal signals.
So a high conduction velocity means that you're receiving the signal faster.
And you're able, it's both sensory and motor output. So both afferent sensory information comes in more quickly, and efferent motor output can be produced more quickly.
Okay, right. So it's just the whole process.
Yep. Yeah, I'll give you some
Signal
examples.
responding. Okay.
Yep. And so this is what it looks like in the cartoon form. And so you've got this myelinated wrapping around an axon. But if you think about it, you need to have those ion channels interacting with the extracellular space from what we talked about before. If you just wrapped the entire neuron, think about wrapping the entire axon, you're not going to be able to convey ions between the extracellular and the intracellular space. And so what you have to do is you have to wrap it, and these are sort of longitudinal sections through a neuron, right? You're seeing the axon here, and you're seeing myelinated wrapping, and then these nodes. And so the nodes of Ranvier are these little nodes A, B, and C. It's called a node of Ranvier, French guy. R-A, right up there, node of Ranvier. And then these are called the internode regions where there's myelin. And so you have the ion transfer happening at the node of Ranvier region, and then in the internodes is where you're moving the ions very quickly. So there's very fast movement of the ions and no exchange with the environment or with the ECF, the extracellular environment. Does that make sense? And so you've got that happening here. This is just showing you a confocal image on the bottom, and then here is a colored colored transmission electron micrograph on the top. And you're seeing this it's basically the same thing is in the cartoon. You're seeing the wrapping, and then you're seeing in the node of Ranvier region, there's these staining for channels, sodium channels. So you see how they kind of overlay with each other. One thing, there's a term, saltatory. So it's saltatory propagation. So you see that the action potentials are being propagated in a saltatory fashion, which just means by jumps. And so you've got these saltatory bits of ion exchange that are causing the action potential to propagate. So if you look at myelin and sort of where it's distributed, at least in the animal world, you're seeing non-myelination in red myelination in red and non-myelination in blue. And so just kind of looking at where this stuff exists, you see myelination in vertebrates, sort of gnathostome vertebrates, jawed vertebrates. You see it in some copepods and krill and mysid shrimp. You don't see it in manna shrimp, which is interesting because they have everything else sensory, but they don't have myelination. Some decapod shrimp, some earthworms, some bamboo worms, and parchment worms. So it's patchily distributed, which is kind of an interesting thing among sort of animals. It's often the case that it's motor neurons that are myelinated so that you can have a quick response. But in the case of the copepod, it's actually sensory and motor.
So just evolutionarily, do you think that they gained it and then lost it, and like why?
Probably gained multiple times. That's the current thought. And then down here, I won't dwell on it, but its conduction velocity is a function of axon diameter. So you see the slopes are all positive for various types of neurons, but you can dig back in. The little thing in the middle is showing you the myelination and non-myelination relationship. So here's V is proportional to D, and here's V is proportional to the square root of D. And so you can just sort of see, if you look at who has really fast but really small diameter axons, mammalian myelinated, for example, that's that curve. If you see who has a really big, fast axon, it's the squid giant axon. These are non-myelinated. If you see who has really big and still fairly slow, it's a lamprey unmyelinated. So you're going to see unmyelinated ones generally having slower conduction velocities, myelinated ones faster conduction velocities, and they can also be smaller. That's just an example of the copepod. We'll skip that. The last two slides deal with information flow at synapses. This is basically how you're taking the production of an action potential, which we just talked about it, and convey it from one cell to the next cell. And you can do that electrically with an electrical synapse. And that's where you would have a gap junction. So you basically have two membranes that are next to each other, and you've got proteins that are spanning both membranes. And so the cells are physically connected. And then you just have ions moving, the sodium ions moving from the presynaptic cell to the postsynaptic cell. And the synapse would be the space between the two cells, right? So in an electrical synapse, you're directly conveying the ions that were depolarizing the presynaptic cell into the postsynaptic cell. Alternatively, you can have a chemical synapse. And at a chemical synapse, you're causing the ions are moving to the end of the presynaptic neuron. They're causing neurotransmitter release. So there's a chemical that's then released, and that chemical is causing a depolarization at the postsynaptic cell or the membrane that it's contacting with. It could be a neuron or a muscle, for example. So the difference here is for electrical synapses, direct conveyance of electrical current. And then in the case of the chemical synapse, you're producing a chemical to be released at the synapse, which is then interacting with the postsynaptic neuron and causing ion channels to open, and then that postsynaptic cell to be depolarized. And then I'll put this up. Sometimes I'll go through this in more detail. I'd rather move on to vision, so I'll just sort of walk through it really briefly. This is showing you at a chemical synapse what actually happens. It's a calcium-mediated process where you've got neurotransmitter, things like dopamine, serotonin, things that you probably have seen in drug commercials, if nowhere else, that are released at the neurotransmitter. So the neurotransmitters are packed up in these little vesicles. And when calcium, basically the action potential arrives at the end of the presynaptic neuron, it causes calcium to enter the cell. That depolarization opens up voltage-gated calcium channels. Calcium moves in, and then these little packets of neurotransmitter fuse to the membrane and dump out the neurotransmitter. That fusion of the vesicle requires calcium, and so you have to let the calcium in for that to happen. So the calcium causes the vesicles to fuse and dump out the neurotransmitter, which are these little blue dots. Neurotransmitter in this case is binding the sodium channels on the postsynaptic membrane, causing a depolarization of that cell, and then you send the action potential down the line. That's an ionotropic receptor. Here's a metabotropic receptor. You're initiating a G protein cascade. So we've already talked about both of these mechanisms at the beginning. And so they can work both the same way. So chemical synapse, chemical information is causing the next round of depolarization of the next cell, as opposed to electrically directly conveying it.
Okay, that was the end of the sensory systems introduction. Remember, this is what you'll be focusing your questions on, and then when we get into class this week, we'll review those questions, and we'll also discuss some new material that I'll present during the class time. Okay, so that is the end of the sensory systems introduction video. See you in class.